Hello, this is a video on DNA structure, DNA replication, and transcription by Selcho, Tristan Diaz, and Jacob Garcia. Today we're going to be going through how DNA uses transcription to turn into mRNA, and how mRNA uses translation to turn into protein.
Rosalind Franklin had a big role in the discovery of the DNA structure. She found that it was in a coiled helix shape by taking X-rays using radiology. She named the photograph 51, and she did this with the help of Maurice Wilkins. In 1953, Watson and Crick discovered chemical structures of DNA with the help of Rosalind Franklin. They later got a Nobel Peace Prize. Covalent bonds are located between sugars and phosphate groups, and between sugars and bases. Hydrogen bonds are located between bases. The stronger of the two are covalent bonds. The beauty of the design is, when the helicase goes through, it is strong enough to break the hydrogen bonds, but not strong enough to break the covalent bonds. Thymine and cytosine are pyrimidines. Adenine and guanine are purines. The complementary bases are A to T and G to C. Thymine to adenine have double bonds, while guanine to cytosine have triple bonds. A nucleotide is the basic building block of DNA. It is made up of a phosphate group, a sugar group, and a nitrogenous base. The phosphate group is on the outside, while the nitrogenous base is on the inside, and they are both connected by covalent bonds to the sugar, which is in between them. The double helix refers to the structure formed by double-stranded molecules of nucleic acids such as DNA. The double helical structure of a nucleic acid complex arises as a consequence of its secondary structure and is a fundamental component in determining its structure. The DNA double helix polymer of a nucleic acid held together by nucleotides which base pairs together. The DNA double helix are complementary strands which run in opposite directions alongside each other. They are two biopolymers which are anti-parallel because they run parallel to each other but with opposite alignments.
This is DNA replication. DNA replication occurs in the nucleus of a cell. This is the helicase. The helicase acts as an unzipper. These are SSB proteins. They separate and keep everything apart. These are free-floating nucleotides in the nucleus. The DNA polymerase is the matchmaker or builder. And the RNA primase tells where to start. This is the ligase. The ligase is like glue. It is the last enzyme. The leading strand is from 3 to 5, and the lagging strand is 5 to 3 prime. Okazaki fragments only are on the lagging strand, and semi-conservable replication copies DNA.
The DNA turns into RNA in order to leave the nucleus. After leaving the nucleus, it goes to the ribosomes. RNA polymerase tells the DNA when to start and stop. This is mRNA. It replicates thymine. Anticodons connect to codons on the DNA. At the end of an anticodon is an amino acid. The tRNA is inside of the ribosomes, giving it the directions to build a protein. The protein is being printed out of the ribosomes in groups of 3 nucleotides called codons.